Hi, welcome to the second part of this lecture on genetics of psychiatric disorders. So in the first part, we talked about genetics of mental illness, and in this second part, I'll be talking more about intellectual disability and dementias. So to begin, there's a whole variety of genetic variation which can give rise to a lower IQ, and I suppose one of the ways to think about it is that in general, there's a view that intelligence can be viewed as a trait that's normally distributed in the population, like a whole lot of other quantitative traits like height and obesity and what have you. So there's a kind of general normal distribution, but then there's a whole bunch of things that can specifically go wrong and that can have a very profound effect, but potentially on reducing somebody's IQ and can make your IQ be much less than you'd be expected according to a normal distribution. So there are some recognized, there's a lot of recognized genetic contributions to that, although it's still the case that in people with profound intellectual disability, we still don't find a cause in sort of around half or somewhat less than half of them. So there's what I'm going to do is take you through a few specific examples, but to say that there are dozens, if not hundreds, of specific syndromes, part of which genetic disorders which can have intellectual disability as part of the phenotype. So some of those microdeletion syndromes commonly cause intellectual disability, but also just point mutations in lots and lots of different genes. And these are sometimes sort of classified as neurodegenerative, syndromic, and non-syndromic. So syndromic means that you've got intellectual disability and some other detectable abnormalities in the phenotype, and non-syndromic means you just have the intellectual disability and otherwise completely normal, apparently. And then neurodegenerative means that the child is born with normal intact cognitive functioning, but then stuff goes wrong and they acquire intellectual disability as whatever the disease processes progresses. So I'm just going to show you a few examples that illustrate specific causes of intellectual disability. And the one that we'll begin with is phenylketonuria, which from a genetics point of view is very interesting in a number of different ways. It illustrates a number of things. So phenylketonuria is kind of a typical Mendelian recessive disorder. So it's caused by mutations in the PAH gene, which codes for phenylalanine hydroxylase, and this metabolizes phenylalanine. And if both copies of that gene are damaged by mutations, so you've got two copies of the gene, if both are damaged, then you don't have functioning phenylalanine hydroxylase, and then you get toxicity from the phenylalanine that's circulating in high levels, which is a normal, phenylalanine is a normal amino acid that's part of our diet that we use to make proteins, but at high levels it's toxic to the brain. So it's very sort of, on one hand, sort of simple recessive disease. It's very kind of straightforward from that point of view. However, it also illustrates it's a good example of a gene-environment interaction, because although it's a Mendelian recessive disease, in fact, you don't develop the phenotype unless you're exposed to phenylalanine in your environment, which here is in your diet. You eat food and it's got phenylalanine in. If children are diagnosed to have that condition, the phenylketonuria, and are given a low phenylalanine diet, so there's not so much phenylalanine in the environment, then they don't develop the phenotype of intellectual disability. So, so you've got a contribution from the genes, which is the mutations in the gene, and you've got a contribution to the environment, which is the phenylalanine in the environment. So it's a very nice example of gene-environment interaction. So that's one thing about it. And now another thing about it is, although it's relatively common for a Mendelian disorder, it's one of the commonest, it's 1 to 10,000 or so live births. In modern Western societies, you never come across it as a phenotype. You never find people who've got phenylketonuria and they've got intellectual disability. And the reason for that is that every baby is screened at birth for phenylketonuria. And so that's what used to be done by a Guthrie test. There's another test used for it now. And so it's a good example of a public health intervention where you have universal screening, and the universal screening is very, very successful. And it means that every child with the condition goes on a low phenylalanine diet and doesn't get the phenotype of phenylketonuria with the intellectual disability as they grow up. And the third thing that's kind of slightly interesting to know about it from a genetic condition is that there's lots and lots of different mutations around which could cause, which will damage the gene and would cause the condition. Meaning that if you were to do genetic testing for this genetic disease, you would have to test for all of those hundreds of mutations in order to make sure that the subject didn't have any of them. Whereas if you just do a simple biochemical test, which is to leave the baby alone for a few days and then measure the phenylalanine in its blood, then you have a biochemical test which is telling you, is that enzyme working properly or not? That's how it's diagnosed. So you have a genetic condition, but actually you're much better off diagnosing it with a biochemical test than with a genetic test. So phenylketonuria, and I hope you see that it exemplifies a few useful key points about genetics and its effect on cognitive function. So another thing to know about is Fragile X syndrome, which is of note because it's the commonest cause, a specific cause of learning disability, intellectual disability in males, and it occurs so that's one in 4,000 live male births, and its cause, it's also interesting that its cause, which is a trinucleotide repeat expansion, so a CGG repeat. So you have a bit on your genome where it goes CGG, 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 and we've all got that, but people with Fragile X syndrome, that the number of repeats that you have is expanded. You have too many of those repeats, and the effect of that is to disrupt the gene which the trinucleotide repeat is in, and that gene was named FMR1, Fragile X mental retardation 1. And then another thing to note about that is that the number of repeats can change in meiosis, so going from one generation to another, and then so what you can have is you can have a pre-mutation where you have more than the normal number of repeats but not enough to cause too much in way of problems in one generation, and then as in the next generation, that number can increase and can cause a problem. So that you can see a pattern of inheritance that could be like this. So it's on the X chromosome. So you might have a grandfather here with a pre-mutation, and so it's not big enough to cause him actual problems. But when he passes it on to his daughter, it gets a little bit longer. But she's also got, because it's on the X chromosome, she's got 2 copies of her FMR1 gene, and the other one is normal, so she doesn't have any phenotype. She's perfectly normal, even though she's got a somewhat expanded trinucleotide. And then here it expands again and goes on to the son, the grandson, and then who has got a longer trinucleotide and repeat and no normal gene to balance it. So it has this interesting pattern of inheritance that you can see. And also of note is that this was one of the first genetic abnormalities causing a disease to be identified, because it produces an abnormality that you can see under the microscope. So it's called Fragile X syndrome because if you culture cells in a low folate medium and look at their X chromosomes, it sort of looks as if the tip of the X chromosome is kind of falling off. And this is actually where the site of the trinucleotide expansion is. And what's happened here is that it's not actually falling off. It's that the chromosome hasn't condensed normally the way it would. And so you're seeing this sort of uncompacted bit of the X chromosome here, which gives it this appearance. So that's Fragile X syndrome, commonest cause of intellectual disability in males, trinucleotide repeat expansion, which disrupts a gene, and observed by microscopy in the first place to identify the location of the gene, and with this pattern of inheritance where you can actually get the repeat expansion increasing down through generations. And what we'll see later on is Huntington. So this mechanism here, where the trinucleotide disrupts the gene, what we'll see in Huntington's is a different mechanism for trinucleotide repeat expansion to cause problems. But here it's just that it makes the FMR1 gene not work properly. And FMR1 we see in schizophrenia seems to be it controls the expression of a number of other genes which seem important for correct neurodevelopment. Just to mention autism, just to say, mainly to say that autism is very genetic. There's a major genetic contribution to autism, and the latest work, what we found, especially from sequencing studies, is that there's a relatively high contribution to autism risk from de novo mutations. And what we're seeing around that, and it's kind of logical if you think about it, is that the more a particular condition reduces your reproductive fitness, the more of it will be contributed to by de novo mutations. In a way, it's kind of obvious. So what we've seen is that if you take the stage from, say, schizophrenia, autism, severe intellectual disability, a lot of severe intellectual disability is caused by de novo mutations. Quite a bit of autism is, and some of schizophrenia is. And what that reflects is that people with schizophrenia tend to have, on average, fewer children than people without. People with autism, fewer still, and people with severe intellectual disability, fewer still. So in a way, if you're going to have a condition occurring in people who are not actually, a genetic condition occurring in people who are not reproducing themselves, then that condition must either be a de novo mutation or it must be a recessive condition where you've got two different sources of disease, or potentially an X-linked recessive condition where you can have unaffected carriers, because otherwise there's nowhere else for the disease to come from. So you know, that's a concept we've just developed over the last sort of 3 or 4 years really, that you see more de novo mutations in the more profoundly your disease affects reproductive fitness. And so that is a moderate effect in autism. So you get some inherited contributions towards autism. We identify a number of genes where there are variants which increase the risk of autism. There's also a polygenic contribution to the risk of autism, which is additive on top of those major gene effects. There's a few specific causes identified. I've mentioned up there mutations in the slide, mutations in the neuroligin genes, and you might recall that neuroligins interact with neurexins in the neural synapse, and the neuroligins are on the postsynaptic membrane. So again, it looks as if autism can be due to disruption of normal synaptic functioning in some sort of way. And the other thing to mention is there are some specific CNVs which increase the risk of autism. In particular, one of the earliest to be identified was placed on 16p, where, interestingly enough, if you have a deletion, you tend to get autism, and if you have a duplication in the same place, that increases your risk of schizophrenia. There's a reciprocal effect on risk of schizophrenia versus autism. Other CNVs, that's not the case. There are some CNVs which increase the risk of autism and of schizophrenia. And there's a number of specific causes which are kind of increasingly being identified. So, and then just to say where we are nowadays in terms of being able to make a genetic diagnosis. So there's if a child presents with developmental disability, including intellectual disability, then there's a number of techniques that can be used. So one can look at the chromosomes themselves, see if there are trisomies, the abnormal number of or partial trisomies, or translocations or other chromosomal abnormalities, just by looking at chromosomes under a microscope, which is called karyotyping. There's a special technique for for detecting copy number variants called a CGH array, although that can also be done by whole genome sequencing. The CNV detection can be done by whole genome sequencing, but you can do a hybridization. CGH stands for hybridization, to detect copy number variants. And if you have a particular syndrome, then you can have you may know a pattern, a whole set of genes which might be responsible for that syndrome, or a whole pattern of variants, and you can search for specific variants with a genotyping panel. If you want to look at particular genes, but you're not sure which variants they might be, then you could do targeted sequencing, although nowadays you probably wouldn't, and you'd move straight to whole exome or whole genome sequencing. And essentially, now the sort of success rate for that eventually to turn up some sort of definitive diagnosis is around sort of 50%. Once that's kind of what's being reported. If you take previously undiagnosed cases, do whole genome sequencing, then you can get a plausible genetic diagnosis about half the time. Okay, so let's move on now towards the dementias. So dementia is a condition where you begin with normal cognitive function, and then you have loss of cognitive function in different areas. So it's not one specific area of functioning. It often begins with loss of short-term memory, but then you have problems in other areas: judgment, decision making, speech, comprehension. And so the notion of dementia is that you have acquired cognitive deficits in more than one realm, and that progresses. And as you're probably aware, it's a very common condition, although I think one thing to emphasize is it is not a part of normal aging. It is not that this happens normally as people get older. This is still something that affects a minority of people as they get older, maybe only around 10% of people, but it is nevertheless clearly, so it's a disease, it's abnormal, but it's also very common. And it's a very, very, very important health problem, both in terms of the disorder itself, which is kind of awful, distressing, debilitating condition for the person who's got it, for the people around them, and also for its effects on society. So a huge amount of healthcare and social care costs go into the fact that people have dementia and the care and resources that are needed to support them, and also indirectly in terms of their carers, the fact that if somebody's caring for somebody with dementia, then they can't work, and that has an economic cost for society as well. So whether you look at it economically, socially, or from sort of a humanitarian point of view, these are really, really important conditions. And and while I'm on it, it's shocking that so little resource has been put into investigating, researching them. I mean, people will say there's a lot going in, but I have to say that proportionally, there's very little has gone into researching dementia compared to what ought to have gone into it. That's been remedied to some extent, but still a way to go, in my opinion. So just to say, so from a genetics point of view, there's a few, or there are some very, very rare dementias which have genetic causes and Mendelian genetic causes, and most of dementia is either Alzheimer's disease or something called multi-infarct dementia. Multi-infarct dementia is you have just like lots of little blockages of the blood cells, the blood vessels in your brain. And so like sometimes people might say mini strokes, but it's like atherosclerosis. It's like the coronary heart disease. It's just you get blockage of blood vessels, and then the brain cells are starved of oxygen, nutrients, and don't work and die. So, and that accounts for sort of about half of late onset dementia. And the genetic contributions to that are mainly probably the same genetic contributions to cardiovascular disease in general. And there's something called CADASIL. There are one or two genetic causes of that, but it's mostly a sort of just the consequence of atherosclerosis. It shares all the risk factors that other cardiovascular conditions do. And then there's Alzheimer's disease, which we'll come to in a bit. So Another one specific neurodegenerative disease, which does have dementia as part of it, arguably dementia as part of phenotype neurocognitive dimension, is Huntington's disease. And this is worth knowing about for a number of relevant reasons. So Huntington's disease is inherited as a Mendelian dominant disorder. And one thing to say about it, so it's characterized by neurocognitive degeneration and also a movement disorder, which is called chorea, which is sort of like jerky movements. And it was originally called Huntington's chorea because of those abnormal movements. But a main feature is also loss of intellectual function. So one of the things about Huntington's disease is that it was one of the first diseases to be mapped by the classical genetic mapping methods that we've talked about earlier in the course, where the approximate localization of the gene was achieved through linkage analysis. So Huntington's disease was segregating pedigree, so you could do linkage analysis, get an approximate localization, and then you got a finer localization by doing linkage disequilibrium mapping, fine mapping, and finding markers that were more and more closely associated with the disease along the chromosome until you had a close chromosomal location for it. Then you could look at genes in that area, and people did this and they found that in a gene there was a trinucleotide repeat expansion, and they named the gene Huntingtin. So again, what you have is we all have a certain number of CAG repeats in our Huntingtin gene, and what goes wrong in Huntington's disease is that one has too many repeats. And what that means is that CAG codes for the amino acid glutamine, and that means that if you have too many CAG repeats, you're making a long polyglutamine tract. And it turns out that cells are not able to properly metabolize the polyglutamine if it's too long, and then by mechanisms which are not fully understood, that there's accumulation of the polyglutamine tracts, and that is somehow toxic for cells, and it's especially toxic for nerve cells, and so you get this neurodegeneration. So this contrasts with the trinucleotide repeat expansion in Fragile X syndrome, which just caused damage to the gene and stopped it working. Here, you've got a sort of gain of function effect. You've got the product of the CAG expansion, which polyglutamine, and it's the product which causes the problem. And it may well be that the Huntingtin gene itself really is pretty irrelevant. It doesn't, what that does, maybe nothing to do with Huntington's disease. The point is that you're making a whole bunch of polyglutamine, and that's cluttering up your brain and giving you the disease. So what that meant was that people could diagnose it, and one of the again important messages to get across about Huntington's, a general message for research, for translation into clinical practice and what have you, is that this discovery was made a very long time ago, I think more than 20 years ago, and it has not turned into a treatment for Huntington's disease. We are just now, not me, but at UCL, and there's one at UCLH, the University College Hospital, they are embarking on a trial of a treatment which is based on this genetic insight, but it's taken this long before, for us still to actually not have a treatment that's available to patients yet. All we have so far are trials. But so, and there's more than one trial going on, but just to mention this as example of the kind of ways that genetic disease can be approached. So what this is a trial of antisense oligonucleotide. So what that means is that you make a bit of DNA or RNA, and you do stuff to it to make it easier to be absorbed and to last longer and so on. But essentially you have a stretch of nucleotides and oligonucleotides, a short number of nucleotides, which match the sequence of a gene that you are trying to repress, to damp down the expression of, and here Huntingtin. So what you do is you have an antisense oligonucleotide for a bit of Huntingtin, or the promoter of Huntingtin, whatever, and the way the trial's working is it's being given into the cerebrospinal fluid via the it's injection into the spine, goes into cerebrospinal fluid, goes into your brain, and it reduces the expression of huntingtin. It reduces the expression of that huntingtin, which is going to be coding for the polyglutamine. And the hope is that you will then make less polyglutamine and cause less damage. You're getting less expression of your huntingtin, but so far as everyone can see so far, it doesn't matter if you damp down the expression of huntingtin. People aren't too sure what it does, but it doesn't seem to be doing anything particularly useful, and people seem to be able to manage perfectly well with their huntingtin expression repressed in this way. So that's nice, and it's a nice example of how antisense technology is being trialed for treatment of genetic diseases in humans. And it's also a nice, not very nice example of how long it takes to go from a very full insight into the pathology of a disease. You can see the variant, you can see its product, you know what's going on, but does that give you a treatment? And the short answer is no. The long answer is, well, maybe in 20 years' time, but it's worth bearing in mind. And then other causes of dementia. So as I mentioned, there's a few rare Mendelian disorders called, and some of them cause a disease called frontotemporal dementia, which particularly attack the frontal lobes and temporal lobes. And so there are identified mutations. One of them in particular genes. I mean, I haven't really got much more to say about that. It's just that one, yes, it has been possible to identify genetic and molecular causes of these rare dementias. It, again, hasn't translated into treatments for them, but maybe we'll see more for Alzheimer's. It allows animal models, it allows one to attempt to start to develop treatments and so on. So now let's talk about Alzheimer's disease, which, as said, is a very, very important public health problem. And so there's a few things to say about it from a clinical point of view. So first off, we're familiar with a very common late onset form of Alzheimer's disease, and there's a moderate genetic contribution to that. It's moderately familial. There is also a form of the disease which acts as an autosomal Mendelian dominant disease. It segregates down through families. 50% of people get it. If you get the mutation, you get the disease. If you don't, you don't. So there's and that has onset much earlier in middle age, in people's 40s and 50s. So the early onset form exists as a Mendelian dominant disorder, and we'll come back to that in a second. And the other thing to say is that people with Down syndrome, which is trisomy 21, develop Alzheimer's disease in middle age, the age of about sort of 50 or 60 or so, kind of universally. That's the thing that happens. So it's all the same disease, and they have a characteristic histopathology, which is that you have, if you in the brain tissue, you have amyloid plaques and neurofibrillary tangles. So the amyloid plaques, so neurofibrillary tangles are neurofibrils that are sort of precipitated, they're tied up inside nerve cells and can be visualized microscopically. Amyloid is an appearance that protein deposits can take up. So if you, there's various diseases that if you have a bunch of one particular protein accumulates in different organs, that can be in your kidney, here in Alzheimer's it's the brain, and you stain it with a stain called Congo red, it has a particular appearance. And so these are microscopic protein deposits. And what happened in Alzheimer's disease is that the protein that made up these protein deposits was named amyloid precursor protein. So the appearance is amyloid, and you can get amyloid from made up from different proteins in different diseases. If you have myeloma, you can get the amyloid made up from the myeloma protein and so on. Here in Alzheimer's disease, it's amyloid, and it's made from amyloid precursor protein. And this is the characteristic hallmark of Alzheimer's disease, and in each of those three forms, the pre-senile, the senile, and the Down syndrome. And what has come to be recognized, it's like these are markers of Alzheimer's disease. It's not that the plaques cause the disease. So there's a poor correlation between the density of amyloid plaques and how severe your dementia is, but it's a sort of part of the disease process. People with Alzheimer's disease have that. And so what people did was they identified 3 genes which cause pre-senile Alzheimer's disease, this Mendelian form. And again, this utilized the classic techniques that we've explained before. So people did linkage mapping in pedigrees, got approximate localizations, looked at genes in those localizations, found variants in those genes. And the first one that was found was APP. So APP gene codes for APP amyloid precursor protein. And it seems so simple now. It was massively difficult at the time. It was identified that there are the particular mutations in the APP gene can cause early onset Alzheimer's disease. Next gene to be identified was a gene, the mutations in a different gene shown to cause other cases of early onset Alzheimer's disease in other pedigrees, and that gene was named presenilin-1. And then people looked at the sequence homology between presenilin-1 and other known human genes, and they found another gene with a similar sort of sequence, and they looked in that gene and they found again mutations in that gene could also cause yet other cases of early onset Alzheimer's disease in other pedigrees, and they named that gene presenilin-2. And then what they did was they showed that the presenilins are involved in processing amyloid precursor protein. So you had a very sort of neat picture, the APP story of Alzheimer's, early onset Alzheimer's disease, which is that if something goes wrong with processing APP, amyloid precursor protein, then you can get Alzheimer's disease. And the ways that can go wrong is that the protein itself starts off being abnormal with an abnormal sequence, or that the enzymes which are supposed to process it are abnormal, they have mutations in them, and that can cause the disease. So a very sort of neat picture, and I still see a little bit of controversy about it today, but I think some of the work was done here at UCL by John Hardy, and well, sorry, John Hardy's at UCL. I'm not sure if he did the work here, but anyway, there's sort of debate in Alzheimer's research a bit about how primary, what's the role for APP? And really these genetic results say, look, one of the ways that you can get Alzheimer's disease is from abnormalities in APP processing. There's no doubt that can give you Alzheimer's, and we know this because if you make a mutation in APP, it can give you Alzheimer's disease. If you make a mutation in the proteins which process APP, you can get Alzheimer's disease. Now, maybe there's other ways to get Alzheimer's disease as well, but it's clearly the case that APP going wrong can give you Alzheimer's disease. So that's early onset Alzheimer's disease, and as far as I'm aware, there's no other known sort of genetic causes of that. There might be some genes that get to be other ways to get it we haven't seen yet, but by and large, presenile Alzheimer's disease is due to mutations in either APP, presenilin-1, or presenilin-2. Late onset disease, those genes are not, well, I was going to say they're not important. Actually, there's a little bit of suggestion that other variants coming out now that in presenilin-2 can influence risk of late onset disease, but basically we'll park that. Late onset Alzheimer's disease, only moderately genetic, not caused by and large by mutations in those other three genes, but with a big effect from allelic variations in a different gene called ApoE, which codes for apolipoprotein E. And the story here is that there are three allelic forms of ApoE, and the commoner form is ApoE is epsilon 3, and the somewhat less common allele is the E4 allele. It's technically epsilon 4 allele, but everyone calls it the E4 allele nowadays, so let's just do that. And the idea is that if you look, the frequency of the E4 allele is commoner in people with Alzheimer's disease. And what happened, this was in 1994, this was, or maybe 93 was the very first paper, but there was a slew of papers that came out all showing very consistently an association between late onset Alzheimer's disease and an increased frequency of the ApoE4 allele. Now, this is what I want to say about this. A couple of things, but I suppose one thing I want to say is this is absolutely unique in terms of association studies for complex disorders. I don't think there's another example of a common variant which has anything like the effect size which ApoE4 does on risk of developing Alzheimer's disease. The effect of the E4 allele is about to double, it's about to triple your risk of developing Alzheimer's disease. So that if you go from having no copies of E4 to 2 copies of E4, then that produces almost 10-fold increase in your risk of developing Alzheimer's disease. Other common variants that we see in genome-wide association studies nowadays produce maybe, if you're lucky, an odds ratio of 1.3 or something. So this odds ratio of between 2 and 3 is absolutely unique for a common variant, and meant also that these association studies of ApoE and Alzheimer's risk were the exception to the rule of that all candidate gene studies are basically wrong. And in what we have, the situation we have at the moment is we've got 20 or 30 years worth of association studies where people looked at disease, they looked at a variant, and they published that was an association between this variant and this disease, and they're all wrong, all of them. There's almost no exceptions, you know, from these candidate gene studies, because they were done on small samples that would only predict big effects, and all those big effects were due to chance. The big effects don't happen in biology because of, you know, selection and what have you. So this is a very kind of unique kind of situation where you have a common variant with a big effect on risk. So there are actually, in fact, 3 allelic forms. There's a somewhat rarer allele called the epsilon 2 allele, and it turns out that that allele is actually somewhat protective. So that the E4 allele increases risk, the E2 allele, you have somewhat reduced risk of Alzheimer's relative to the most common E3 allele. So another thing to say about that is there's a sort of, it was recognized fairly early on that apolipoprotein E does interact with APP. And so there's a sort of notion of some sort of biological plausibility that maybe the way that ApoE was exerting its effect was through its interaction with APP. So this was please don't show this slide anywhere else, it's kind of embarrassingly naive, but the idea was that there could be something abnormal, this is the APP, abnormal about the APP itself, which makes it form more likely to form amyloid, although it's probably not the amyloid that caused the damage, but something about how it's processed, or there's something wrong, something about how ApoE4 interacts with the APP, which makes it more likely to go down that pathway of causing disease. Now, since then, there have been genome-wide association studies and latterly sequencing studies as well, which are sort of throwing more light on the genetics of late onset Alzheimer's disease and showing that variation in other genes can also affect risk of disease. But I think the message to get across is that ApoE is the main one. There'll be other stuff as well, and that other stuff might be very helpful in terms of helping us with the biology, but the main genetic risk for late onset Alzheimer's disease is ApoE4. So one of the interesting genes was this one, SORL1, which is sortilin something 1, sortilin related protein 1, something like that, which again is kind of which is related to the processing of APP. So here's a more sophisticated diagram of the processing of APP, which shows you where some of these important enzymes come in. So the presenilins are involved here in basically the APP protein can be cleaved in sort of different kind of ways. Then SRL1 determines whether it goes into the base, which is a enzyme which cleaves APP, and what you don't want happening if you don't want to get sorry, the presenilins come into this bit. If you don't want to get Alzheimer's, what you don't want to get made is the A beta fragment. So APP can be metabolized in a number of different ways, and if the way that it gets metabolized ends up making a lot of A beta, then that is the A beta fragment of the APP, then that is the thing that will give you the increased risk of Alzheimer's. And so we could see that sortilin is kind of up here, and it does seem that variants in this gene can affect disease risk to some extent. In the presenilins, certainly for the early onset disease, although it's looking like also some extent in the late onset disease as well. And also, as I say, apolipoprotein E. So what have we got from that? Again, a bit like the Huntington's story. So these Alzheimer's genes were discovered decades ago, and yet, you know, we don't have any effective treatments for Alzheimer's. We don't have ways that interfere with this process. What they have allowed us to do is to make animal models of Alzheimer's disease. So you can make mouse with Alzheimer's disease. You can think about things that might work, and you can try giving those things to mice, and you can see that the mice, you know, the disease process gets halted or reversed, and that's all fine. But what this field has been spectacularly unsuccessful at so far is translating those insights from the original genetic studies and from the animal models into successful drug targets. So, you know, this week, as I speak, we have seen an antibody treatment which is directed against the APP. So the idea is that it kind of mops up the APP or the A beta before it has time to do too much damage. And essentially, there's been this huge drug trial, and although there's a bit of controversy about it, I think a lot of the statistical experts are saying that they find that drug trial very sort of unconvincing. The drug is being considered by the FDA, I think, as I speak. I think they haven't quite decided yet. But here we are a long, long way down the road, and with no drug treatments which really make an important impact on the progression of Alzheimer's disease, in spite of the fact that we were, you know, been aware of a lot of the core biology for a very long time. We've had mice that we could study it in, and, you know, we are where we are. Okay, well, that's really all I wanted to say about the intellectual disability and dementia genetics aspects. I hope that's been useful. These slides are going to be available online, and if this were a live lecture, I'd be asking for questions now, but it isn't. But I hope that's been useful anyhow, and good luck with your studies.